Okay, hi guys. So today myself, Dana, and Maddie will be talking about crown illumination as a predictor of tropical tree growth. We posed two ecological questions, the first of which being, is crown illumination index a predictor of tree growth? Crown illumination index is a categorical variable with 6 codes for the various levels of sunlight. These values were reported as a mean of 2 measurements, which allowed us to treat them as continuous with intermediate values represented. We predicted a lower height and greater diameter because with higher crown illumination, trees don't have to grow upwards as far to reach the sunlight, and they can focus their energy on growing wider, whereas in the shade, they will reach taller in order to find the sun. The second ecological question we were asking was, does crown illumination index depend on species? And we predicted that higher crown illumination index will lead to higher annual diameter increments and lower annual height increments for shade intolerant species, and the opposite for shade tolerant species. That prediction is based on the observation that shade tolerant climax species tend to shoot up to fill a gap in the canopy when it opens up, and that shade intolerant species lacking sunlight tend to grow tall towards the sun. Next, just a little background on the La Selva biological station where the study was done. So it is in a tropical wet forest in Costa Rica. There's a large diversity of different habitats from elevation differences and drainage patterns. There are 3 rivers that border the station, so it's well irrigated. Some natural disturbances have been recorded by indigenous peoples around 1,000 to 2,000 years ago, so it has been a disturbed area for a long time. Holdridge acquired the core piece of land in 1953, and he set up a lot of research opportunities in the area, and it was basically a full research station by the 1980s. So then for the field materials and methods used by the researchers, they were focused on annual accurate long-term assessment, so making sure that the same tree was recorded every year. They were looking at specifically the survivorship. So if the tree had been damaged in any way, they would take a note of that. The growth, height, and diameter, as we discussed before, was also taken note of, and the total microsite conditions. So that includes the crown illumination, as we were talking about, the number of overtopping crowns, or the local forest structure.
Okay, so the data set we were working with contained information for 10 different species across seven separate data files, and some of those had species codes and some were missing. So we had to match the species codes to unique tree identifiers for the growth and microsite data files, referring to the individual's data file. We then joined the growth and microsite data files by tree identifier in R. We made subsets of the joined dataset by species code in R, and then we were able to conduct our 2 multiple regressions, testing for an association first between height species and crown illumination index, and then diameter species and crown illumination index. So here are the results for the multiple regression for diameter. So we're looking for large F values and low P values to reject our 3 null hypotheses. So we were able to do that. So we could reject that there's no variation in diameter among species, no variation in diameter with changing crown illumination index, and no variation in diameter among species with changing crown illumination index. So we made graphs for each growth parameter versus crown illumination index with our species grouped by similar average crown illumination index. For this first graph here, we can see a weak negative relationship in blue, and then otherwise there are positive relationships of varying strength. For the next 2 graphs for diameter versus crown illumination index for the remaining species, they all have positive relationships that again vary in strength. So here are the results of our multiple regression for height. Again, we can reject the 3 null hypotheses that there's no variation in height among species, no variation in height with changing crown illumination index, and no variation in height among species with changing crown illumination index. So our graphs for height versus crown illumination index showed similar trends. So we've only included one here for 4 similar species. So we saw positive relationships varying in strength across all 10 species.
So one of our key findings was that species one, the Cesropia obtusifolia, which is pictured on the right, was the only species with a negative relationship between diameter and crown illumination. This was not entirely unexpected, as this species is the one with the smallest maximum diameter, almost of 37 centimeters, almost half of the next species with the smallest trunk of 67 centimeters. We knew the relationship between the diameter and crown illumination thus would be a weak one. However, we did not anticipate it to be negative. Since the negative relationship was weak, it could simply be due to observational errors. In the metadata, our authors spoke regularly of measurement errors that had to be double checked, so this could be a possibility. However, we do also know that this species is a fast growing but short lived species that needs full sun exposure to thrive. This could attribute to its negative relationship, as the greater crown illumination, the faster the tree shoots upward, simply not allowing diameter to keep pace with its height growth, forming a compact trunk with weak and soft wood prone to rotting. Height or length is a process that comes from primary growth, while diameter or girth comes from secondary growth. As such, in order to grow laterally, a plant will need a more complex and robust vascular system that takes longer to produce. Since it is a short-lived plant, it may not have the time to develop such systems to match the primary growth. This could lend reason to why the structure of the wood limits the species' height capacity. Another key result was that for all 10 species, we were able to find a positive relationship between height and crown illumination. This result was partially similar to that which was found in other studies in the field, such as Sturck and Bonger's 2001 study on crown development in tropical rainforest trees, where they studied patterns with tree height and light availability. Here they found that height growth seemed to be controlled by light exposure, thus have a positive relationship in trees up to 25 meters in height. Since the average tree size in our study was approximately 3 meters in height, this pattern would also apply here. However, in this study, the authors also found that as trees became taller, the relationship became nonlinear. This pattern was not seen in our study. However, this could be attributed to the length of the study. Although a 34-year-long study does sound relatively lengthy, in regards to the average lifespan of a tropical tree, it is actually quite short, as they can live for hundreds of years. Perhaps if the study continued for longer, our results would begin to show this nonlinear relationship. This study did caution that changes in height might not have to do with higher light levels, and thus looking at other factors which contribute to growth, like nutrition levels and water abundance, would be beneficial for assessing the confounding variables. We had two main conclusions in our study. The first answered the question if crown illumination index is a predictor of tree growth with respect to height and diameter. Our results were able to support the prediction that diameter increases with crown illumination. However, it was not able to support the prediction that height decreases with an increase in crown illumination, and thus the prediction was refuted. Instead of finding a negative relationship, we found a positive one, as mentioned previously.
To answer our second question, we were able to conclude that growth varies differently with crown illumination index among species, but our more specific predictions regarding higher annual diameter and lower annual height for shade intolerant species versus the opposite for shade tolerant species were not supported. Apart from species 1, as we previously discussed, all species, independent of whether they are shade tolerant or shade intolerant, had a positive relationship between diameter and crown illumination index. The relationship between height and crown illumination index for all species was positive as well. However, a qualitative analysis of the graphs produced showed that generally, the strongest relationship between growth and crown illumination index was for a shade intolerant species, and the weakest relationship was for a shade tolerant species. This preliminary analysis needs statistical testing to determine its significance, which we plan on conducting for our report.
For future implications, understanding the information from our experiment can allow for three things. One, better understanding of which species are able to coexist, as it aids in a more robust understanding of niche differentiation with respect to their light requirements. Secondly, it can aid in economic endeavors, specifically the produce market, as crown illumination is one of the predictors of fruit maturation in tropical dioecious trees. And thirdly, Understanding how crown illumination affects the growth of tropical trees gives aid in the fight to combat climate change, as tropical forests constitute a major terrestrial carbon sink. Thus, finding the optimal growth for tropical trees and carbon consumption is an apparent step towards a positive climate change.
Okay, and that is everything. Thank you guys.